Hello and welcome to our 17th Virtual Research Cafe. I'm Frank Gomez at the Chancellor's Office, Executive Director of STEMnet. Behind the scenes, we have Monica Alacon, Operations Analyst for STEMnet. Much thanks to her for doing a lot of the background and logistics for today's event. The goal of the cafe is to help foster collaborations across the CSU and perhaps catalyze the submission of a proposal later in the year. We call it 10.0 because each of the speakers will have 10 minutes each to speak. We hope you'll find the opportunities for collaboration fulfilling or maybe actually open a new area of research for you and a collaborator downstream. Please feel free to contact any presenter in the chat box when they're not presenting. At the end of all three presentations, we'll all come together and there'll be a Q&A session for everybody to ask questions of the speakers and even for speakers to ask each other. So let's begin. Our first speaker is from CSU Dominguez Hills, Samantha Leigh. Samantha, or perhaps it's Sam, the floor is yours.
All right, thank you so much. Let me go ahead and start sharing my screen quick. All right, can everyone hear me okay? Okay, great. So thank you so much for being here. I'm an assistant professor finishing up my second year at Cal State Dominguez Hills. I'm very excited to speak with you all today about some of the research I have going on. So I'm a nutritional physiologist and I mainly focus on marine organisms. My area of expertise is on what fishes eat and how they process that food that they eat. What an animal consumes is arguably one of the most intimate interactions it can have with its environment, since it's taking something from outside of its body, consuming it, putting it inside its body, and then of course also breaking down some of those components and excreting them back to the environment. The efficiency with which they do this is also extremely important, so that they have nutrients necessary for functions such as movement, growth, reproduction, and so on and so forth. Fishes in particular represent really great systems to study questions related to nutrition and digestion because they make up one of the most diverse groups of vertebrates on the planet. To our knowledge, there are over 34,000 different species of fish, and we're discovering more and more all the time. They consume an extremely vast variety of food types. There are fish, just to name a couple of the ones up here, like the whale shark, biggest fish in the ocean that consumes plankton, fish that feed on mollusks, other fish, even catfish that eat wood. If you can think of it, there's probably a fish out there that eats it. And of course, as humans, we depend on fish for food and economic resources. In California, over 120,000 people are employed by the seafood industry, and it generates over $200 million per year to the state's economy. Even more on a global scale, over 3.5 billion people worldwide actually rely on seafood as their primary source of protein. So it's really important to understand how fishes fit into ecosystem dynamics. Unfortunately, not everything that marine organisms are eating these days is actually food. I'm really interested these days in understanding how marine animals are able to deal with synthetic material that they ingest, mainly microplastics. So microplastics are plastic particles, fragments, or fibers that are less than 5 millimeters in diameter. And they get into the ocean in a couple of ways. So one option is that macroplastics like water bottles, tires, forgotten fishing nets, things like that, degrade over time due to weathering and sun damage and create really teeny tiny pieces. There are even microplastic fibers that come off of our synthetic clothing every time we wash them. We also intentionally manufacture plastics to be small, such as microbeads in cosmetics, microbeads for numerous industrial purposes, things like glitter. And all of these fibers and particles find their way through water treatment, through stormwater runoff, and into our waterways and ultimately the ocean. Unfortunately, this means that these plastics are entrapping as well as being ingested by organisms at all trophic levels, even as small as these plankton here that I collected off the coast of Catalina Island. So what I want to figure out is pretty much how pervasive microplastics are, where we might find them, as well as what adverse effects they could be causing. So thus far, my students and I have collected water samples, plankton samples, as well as tissue samples from 2 commercially important fishes, anchovies and California halibut. And we looked at samples from different habitat types in the Southern California Bight. So everything from LA Harbor, very heavily trafficked port, actually the most heavily trafficked port in the US, all the way to the west side of Catalina Island, where obviously there's going to be a lot less human traffic. Unfortunately, we did find microplastics in every single sample from every single location. And this is a map that was created by master student Chelsea Bowers, who I actually worked with at Cal State Fullerton, showing distribution of plastic, either particles or fibers, from the different locations we sampled for both water and plankton. What we found was that the majority of microplastics that we found, at least in these surface waters, were fibers. And this could be because those fibers are more positively buoyant. So if we were to collect samples kind of throughout the water column, not just at the surface, but more the neutrally buoyant and also at the benthos, we would probably find different types and amounts of plastic. And one of the most alarming kind of finds from all of this information was that LA Harbor alone contains about 275,000 microplastics per meter cubed in surface waters. So to put this into perspective, this was by far the most polluted site of the ones we sampled, but also far exceeds global estimates. So globally, the average estimate for microplastics in surface water is about 102,000. So this more than doubles that. And far exceeds what has been found in other heavily trafficked areas like San Francisco Bay, Chesapeake Bay, the Great Lakes, and even some ports in China and India that have been sampled. We've also shown that marine zooplankton are ingesting microplastics, primarily in the 63 to 125 micron range. So this is definitely a path where microplastics could enter the food web and transfer up trophic levels, affecting those commercially important fishes that we rely on. So currently I'm building on this work by using FTIR, or Fourier Transform Infrared Spectroscopy, to determine the polymer types represented in our samples so that we can begin to get at the primary sources of these microplastics. So for example, are these fibers mainly coming from tires or synthetic clothing, or is it something else entirely? Additionally, I mentioned that we have collected 2 commercially important fishes, the anchovy and California halibut, and we're looking at their various tissue types, including the muscle tissue that humans would consume, to figure out the extent to which microplastics are found. This data is still being analyzed, but yet again, we are finding microplastics pretty much in every single sample. And now that we've established that microplastics are very pervasive and we have a better idea of what types of concentrations fishes are exposed to in the wild, we're going to do some in-lab experiments using zebrafish as a model organism. So my master student, Will, is working on this, and he's going to expose them to the concentrations we found in the samples that we sampled in LA Harbor. And we'll see how things like their growth, fecundity, and other digestive parameters could be affected. So this type of work is really critical to understand the potentially detrimental effects of microplastic pollution in the marine environment, but it does not stop the further contamination of these critical species and habitats. The world has seen an average of a 68% drop in overall animal populations since 1970, so not even that long ago. The biggest drivers of this immense loss of biodiversity in recent years are thought to be habitat destruction and pollution. So we need to find a way to prevent microplastics from making their way into the oceans in the first place. So 2 of the ways that microplastics make their way into the ocean are through wastewater treatment and stormwater runoff. For wastewater treatment, there are 3 general stages of filtration. So in the primary stage, materials settle to the bottom. This is called sludge, and are removed, along with floating materials on top kind of being skimmed off the surface. The remaining water consists of suspended materials and undergo the second treatment stage. And during this stage, those neutrally buoyant particles flow through these aeration tanks that are filled with microorganisms to break down organic materials. And then filtered water then moves into the tertiary stage where it's going to be disinfected either with ultraviolet rays or mixed with some sort of disinfectant to kill any harmful bacteria, viruses, or other microorganisms. So obviously something that is lacking is some sort of mechanism to target the removal of micro or even nanoplastics from the water, particularly neutrally buoyant ones. With respect to stormwater runoff, there really is no filtration of that material whatsoever. So really what's needed is some sort of filter that's capable of removing these plastics from the water before it gets to the ocean. Looking to nature for inspiration has obviously led to many diverse technological advances, and seeing as I studied digestion, I looked to the digestive system for inspiration, specifically in an organism that has existed since before the dinosaurs, and that is sharks. So all sharks have a unique segment of their digestive system called a spiral valve, kind of a spiral staircase-like structure in the mid-region of their gut. And I have a bunch of CT scans of different spiral intestines in these different morphologies from over 32 different shark species. And here's just another example of kind of a cross-section of one of these shark spiral intestines. And what I'm planning to do is actually start this summer, 3D print some of these models from these shark spiral intestines and put them in a flume and do some particle image velocimetry or PIV analyses to see how different size particles flow through these really unique kind of natural filter structures that show up in the shark digestive system. And I'm out of time, but I'd like to thank members of my lab and other funding sources and people who have helped support this work. And thank you all for being here, and I'll take questions at the end of the other presenters. Thank you.
Thank you, Samantha, for a very interesting talk. And questions will be taken when everyone has completed their presentations. Our next presenter is Joyce Pham from CSU San Bernardino. Joyce, the floor is yours.
Okay, let me see. Let me make sure that you can see me. Hold on. All right. Okay, so hi everyone. I divided my research group into kind of 4 main aspects of this tetrahedron that you see here. So hold on, my slides are not moving. Okay, we can start first with synthesizing a compound and then determining the structure. Where are the atoms in the structure that we see? And then our next question is really trying to rationalize, well, why are the atoms where they are, and using chemistry concepts? And this kind of 3 facets are all connected to what we call physical and chemical properties that you and I exploit. And I'll talk a little bit about that exploitation in a little bit. But the goal of my research group is really then try to find the common principles that unite these tenets of material sciences. And so I'm going to provide an example here in a little bit about how crystal structures, where the atoms can be related to some of the theory behind why they are there. And if I may, I would say that this research really can be divided into kind of the interface of these 4 disciplines. So crystallography, trying to solve the crystal structure, physics, measuring transport properties, materials engineering, designing materials for specific purposes, and then chemistry is really understanding the relationship then between crystallography and chemistry concepts. And so earlier I said something about exploiting the kind of relationships for our use. And so here's some example of that. So I think most of you probably have some kind of material that you're using that has batteries. And at the fundamental structural level, batteries, at least for lithium ion batteries, solid state batteries, work such that there are layers of atoms. So here you can see examples of layers of atoms with lithium, which is a very small ion intercalated between those layers. And so structurally, fundamentally, where the atoms are affect what we use on a daily basis, for instance, as a battery. Here is another example. So solids that are crystallizing in cages. So here, these are cages, very large cages with atoms that are in the middle rattling, have been studied for instance, thermoelectric materials. So thermoelectric material convert a heat gradient into electricity. So you and I right now are emitting heat, and imagine if that heat can then be garnished so that we can then use it for electricity. And so this kind of physics and materials engineering is one side of this kind of relationship. And I'm focused more at this point on the crystallography and the chemistry side. So understanding where the atoms are is really important because ultimately it affects what we use on a daily basis. And more importantly, we want to understand, well, how can then we tune and get to where the atoms are based on some concepts of electronic interaction. Electrons in various orbitals, how do they interact? Okay, so if you look at the periodic table, you see here that the majority of it is metals. So my group, what we do a lot is we deal with metal-rich compounds or intermetallics. So two or more metals combined at specific molar ratio. And what we do is we cut up these elements and we get products that are crystalline, where we can then characterize, so determine where the atoms are using X-ray diffraction and even single crystal X-ray diffraction. So both powder and single. And so these are data from our actual research group that you see here. And so from then what we do is, okay, we then try and rationalize, okay, let's solve the structure. This is where our atoms are. This is just a cartoon model of what our products actually look like. And then we take a step further, and this is actually using resources of UC San Diego to calculate the electronic interaction, which means electrons reside in various orbitals. How are they interacting with each other such that we can rationalize the formation of the structure that we see? And then from then we kind of go back and fine tune our synthesis or update our project. So there are certain points in this kind of general procedure that you can get involved in if you are interested in collaborating. So for instance, if your group do a lot of synthesis and you're trying to find new characterization tool, whether via powder, electron diffraction, or single crystal X-ray diffraction, please feel free to shoot me an email. And we are installing our first single crystal XRD at CSUSB, so I'm really excited for that. So send your samples my way. If you have a structure that is rather complex and you're trying to find another perspective on it, please also feel free to reach out. I'm happy to provide another insight onto solving your crystal structure. If you are interested in determining how we can rationalize the structures that we observe, I am happy to also receive your data and then calculate models for a way to rationalize that. And this is already in collaboration with UC San Diego. And last but not least, if you're more on the physics and materials side and you enjoy measuring, for instance, transport, electronic transport properties, feel free to also reach out. Okay, so let's kind of take a step back and go into this periodic table again. And so here I mentioned that the majority of it is metals, and I'm highlighting what are called the transition metals right here. They sit right in the middle, and you can see that I've kind of color coded. We have the red, we have the green, and we have the yellow. And actually, those have certain structures associated with them at ambient condition. And you can see that same trend in iron, cobalt, and nickel. So red, green, yellow, okay? And so these structures are in ambient condition, they're body-centered cubic. So what you see is one atom that sits right in the body is also observed in that same corner, another atom, okay? So body-centered cubic. And we have hexagonally close packed and face-centered cubic. And then these metals right here that are called the 3D transition metals, you might know them as magnetic metals, also obey that. So red, green, yellow. But manganese is kind of an oddball, and that is very complex. It has 58 atoms per unit cell in ambient conditions. So my research group, at least for one of our projects now, really try to understand the role of this manganese in this kind of the 3D transition metal. And so this is just one example of the type of research that we do. And so in a collaboration with a group abroad, they found that nickel and zinc crystallize in a structure that is known. So here's the structure that you see. It's a body-centered cubic. So anything that you see right in the middle is also observed right at the very corner. So it's in a series of concentric shells. So in the innermost shell, we have an inner tetrahedron, then an outer tetrahedron, and then an octahedron, and then I'm highlighting here a cube octahedron. So you can also see on the left here, tetrahedron, outer tetrahedron, octahedron, and cube octahedron. And then it's a body-centered cubic. So this is all that is shown. And this structure is known, but what we did experimentally is that we included manganese. So now with manganese addition to the unit cell, we see that manganese prefers to occupy only certain region of the unit cell. So if you add a little bit of manganese, so about 0.5 to 0.6 molar manganese substituting, it actually goes in only the octahedron site. So this octahedron site right here. And it will be in competition with zinc. And then if you add more manganese, so as we add more manganese to a unit cell, we synthetically introduce manganese and then we solve the crystal structure, then we see that it actually substitutes, but it does not go into the same octahedral site. In fact, what it does is that it goes into the inner tetrahedron. So you see this inner tetrahedron right here. So manganese doesn't ever fill up this site. It goes into a completely different site. And so our question really then becomes, why is that? Why is it that when we introduce more manganese, we see this kind of observation? And so we use what's called electronic structure theory to get to understand this. Okay, and so this work was done during the pandemic. So this is my second year. All the work here is from the first year, and my student Mayra Silva Barcenas actually did this research. I think my computer is frozen. Okay, hold on. Yeah, it's not responding. Okay, wait. Okay, so Mayra did these calculations using resources from UCSD, and what we find is that with a little bit of manganese and then a little bit more manganese, these are the only options that manganese can occupy. And so what we find is that both trans arrangement of manganese and mer arrangement of manganese are preferred over Fac or cis, which means that in this atomic arrangement, manganese atoms prefer to be farthest apart from each other. And so now we can also look at, well, how are electrons in various orbitals interacting among one another? And so again, that is using resources of PRP. And so here you see what's considered kind of like a molecular orbital theory, but for solids, we have density of states here and energy, and there's this line that separates from the filled orbitals from the unfilled orbitals. And this kind of calculations then allow for us to realize that manganese substitute for zinc in the structure, but is actually stabilized by manganese 3D, nickel 3D interactions. So now we can kind of go back to this whole kind of general procedure and see that there's this structure that we see, and our collaborator reach out and we're trying to understand why do we see the structure that we see. And so that's just one example of what we can do to really relate this whole scheme of the periodic table in our solid state chemistry. So if you have any questions, I'm happy to answer them later, or feel free to reach out. Thank you.
Thank you, Joyce, and I'm glad your computer was able to come back to life.
Thanks.
Our next speaker is Wang from Cal Poly SLO from the Department of Civil and Environmental Engineering. Long.
Okay, thank you. Oh, my computer will survive. So, hello everybody. My name is Wang. I'm from Cal Poly Civil and Environmental Engineering Department. So the topic of today's talk is going to be on sensing nanocomposites for both structural applications and wearable applications. And just to kind of give a quick intro about myself. I also joined joined my current institute 2020 during COVID year. So it has been fascinating to kind of work with my students on different topics of research, because for myself, my research interests are quite diverse, but the main theme is basically developing new materials for both mechanical and sensing applications. And in between, the material development and applications, we use different emerging technologies to characterize the materials, to understand the materials better, so that we can have a more sustainable framework to develop new other different types of materials. And for today's talk, I will mainly focus on the, like I mentioned, the sensing nanocomposites aspect. So to begin with, I think all of us are familiar with different kinds of sensors in different ways. Here, I basically demonstrated two different kinds of, two different domains of sensors. The one on the left is basically the sensors that can be used on structures, on engineered structures, right? So including all of these, for example, accelerometers, corrosion sensors, etc. So the main goal is to use these sensors to measure the structural response of those engineered structures, apparently. And then on the figure on the right, you can see is more probably for general folks is more relatable, because these are the wearable sensors that are currently on the market. We use these wearable sensors for monitoring our health, right? Typically, or movements and stuff like that. So even though these sensors might take various different forms, they share the same goal, which is to provide rich data on the performance of these different structures. I consider both as structures because it's just some sort of mechanical system that with different purposes, either for engineered structures or for biological system. And then, even though we have all these commercial devices, they also share some of the major limitations. For instance, they are all discrete devices, meaning that they can only measure the structural performance at the location where they're instrumented. And they have limited sensory modalities. So essentially, the sensors are streamlined for one particular parameter measurement. So there's no so-called multifunctionalities inbuilt in these sensors. And power consumptions always limits the long-term applications of these sensing devices. And I would say one of the most fundamental limitations of the existing sensing devices is that they rely heavily on conventional sensing materials, including metals or semiconductors. So the researchers like myself, we basically look into how do we really enhance the sensing materials property by using different kinds of materials. And for my research, I'm interested in utilizing different nanomaterials for developing the sensing components of those devices. For instance, I've used carbon nanotubes and graphene because they have extraordinary electrical and mechanical properties. And I use additive manufacturing approaches to incorporate these nanomaterials into a scalable functional material system. The reason, well, when it comes to scalability and manufacturing aspect of these nanomaterials, one of the major challenges is that these materials tend to agglomerate, basically forming this sort of inhomogeneous feature in a larger material setting. And these inhomogeneous feature or agglomeration will compromise the overall material property and performance. So then it leads to several typical research questions that I am interested in. Basically, how do we design the sensing materials in an efficient way? And how do we use them? And more importantly, whether or not they can perform in practice compared to whether or not they can perform better even than the conventional sensing materials. So in today's talk, I will just highlight, I guess, two different kinds of manufacturing techniques that I have developed before. Each of them have their unique advantages. So the first one is called spray coating. Essentially, is advantageous for large-scale manufacturing of these materials, and is typically can be, I guess it can typically be used for infrastructure monitoring. And in terms of the procedure, essentially I would make an ink out of, for example, the carbon nanotubes, and then we could either manually airbrush this ink onto the substrate of interest or structural surface, or we can use some sort of robotic system to more scalably spray coat this material on the structures. And then this is the SEM image, scanning electron microscopic image of the microstructure of these materials. Basically, you can consider this as some sort of more like a spaghetti dispersion inside of this material. Another type of manufacturing technique that I have developed is basically like an inkjet printing. Is very advantageous for high precision and multi-material patterning. So in this case, I developed based on the graphene, and the setup is essentially like a dispensing needle with very high precision to form the pattern that we can pre-design. And that is an example of a very simple pattern that's on a flexible medical tape that's patterned using the graphene ink. So moving on, once we manufacture the material, like I said, we have to characterize its sensing property. And we are interested in the strain sensing property, and we conduct these electromechanical testing. So essentially, we could apply mechanical load on the material and simultaneously measure the electrical property of those materials. And this is just a figure showing that some of the results that we have, essentially, the electrical property, the electrical resistance is represented by the red line, which is in accordance with the applied strain, which is in the dashed line. This basically indicates that our material can be used for measuring the strains. To simplify the mechanism of this sensing phenomenon, essentially, we can consider this material system get deformed, and then the electrical property will change correspondingly. So in terms of the application, so I will highlight some of the different scope of applications. The first one is on the in terms of the structural health monitoring. So this is the previous test I have conducted. Essentially, I fabricated the material, or I spray coat the material, to be particular, onto a reinforced concrete structural surface. And my collaborator was interested in the seismic performance of this reinforced concrete shear wall. And my goal was to use my sensing material to investigate whether or not we can capture some of the potential damages. And at the end of the loading test, essentially, we were able to introduce the structural damage, apparently. But then what I was interested in was looking at this whole sensing system, whether or not we capture that potential structural damage, or like a structural or a stress or strain concentration. So this is the electrical imaging algorithm that we have to essentially kind of characterize the electrical property of this sensing material, and which can be correlated to the mechanical response of the structure. And the highlight is the hotspot that developed at the end of the experiment indicates that there could be either non-visible structural damage or stress concentration that can potentially propagate into structural damage. So then I will trespass to a different domain of application, which is, like I mentioned in the beginning, the wearable sensing capability of these materials. Because these materials are very flexible, so we could incorporate these materials with different wearable platforms. For instance, this is an example with integration with fabric, so that we can use this type of chest band to monitor respiration, motions, and I have also used these wearable devices for monitoring pulse, etc. And then another device development that I have done before was to use these fabric sensors to measure some of the pressure levels that are applied on the socket prosthesis interface to, how to play this? Okay, this is blocking me. Okay, there we go. we can either measure more controlled level of pressure or a more dynamic version of it while the amputee walks. So similarly, we could incorporate the sensing materials with medical tape and forming some sort of sensing skin, which can be used for human-machine interface or again for prosthetics development. So in terms of with that, I will just quickly acknowledge my collaborators, my students, and my funding agencies, so that I can answer some other questions when we at the Q&A session. I'll leave it like that. Thank you.
Great, thanks, Long. And now we will, very interesting talks, and perhaps subconsciously actually asked people who had materials science work, just somehow in my brain, I kind of went towards 3 speakers that were doing very, very interesting materials work. So we now open it up to questions from anyone and everyone in the audience. You can just either put your hand up or show your video or however you want to ask questions. Patrick, you have a question?
Yeah, thank you. I'm Patrick Wang. I'm at Cal State East Bay. I'm an electronic structure theorist in the chemistry department. My question is for Joyce. You mentioned intermetallics, which are very interesting. And it looks like you're doing some work on magnesium zinc intermetallics. I'm just wondering if it looks like that's wrapping up, and I'm wondering where you plan to take that afterwards.
Yeah, so that's with manganese, manganese, zinc, and nickel. And so that one is being submitted. The actual experimental portion of that is with the collaborator, and we were only doing the electronic structural part. And so I think the next step is trying to add more manganese into the system and see if there are more preferential order. Right now, the lower end of adding the amount of manganese into the system shows that there's a preferential substitution electronically using electronic structure theory in the octahedron and the inner tetrahedron. But what if we were to add more so that it's even more manganese rich? That seems to be the direction that it's going toward.
Thank you.
Thanks for your interest.
Yeah, I was just wondering, were you looking to, it looks like you're doing mainly 3D transition metal intermetallics. Is there some technical barrier for looking at heavier elements, in particular like rare earth? On the other hand, the structures are much more complicated, I understand that. But there's also could be more potentially interesting, phenomena to look for.
Absolutely, yeah, there is no barrier in terms of that. So far, we started on the 3D because they're magnetic, but also you see that as well in the rare earth. There's no barrier in that way. Thank you. Yeah.
Thank you.
I think you raised your hand, Joyce.
Yes, I lowered it. I have a question for Long. So you were talking about characterization using mostly the properties in terms of the strength. I think you showed like an apparatus that you use. Do you characterize any other structural features for your materials, or is it?
Yes, thank you for your question. We do. I showed that we use SEM, but we also use different like XRD, Raman spectroscopy, and even FTIR. So essentially, we do dive deeper in terms of the material characterization aspect, and we can even another interest of ours was to look at the microstructural change under loading, so that we can basically have experiments to instruct the potential simulations to even model this material behavior. So yes, the short answer is yes, we do.
Thank you.
Thank you.
And there's a question for, oops, I think Samantha already answered the question for Mike. In the chat. Okay, great. I have a question actually for Samantha. You mentioned that many of the materials were fibrous in nature, as I recall.
Yes.
And so from a biological perspective, if people were, and I assume they do eat these fishes, where might some of these fibers actually show up in terms of their organs? Do they pass the blood-brain barrier? What's the thinking from the, say, medical field?
Yeah, so there's honestly very limited studies that have looked at this specifically in fishes. There is support for the fact that particles at least are able to translocate from the gut into the blood tissue. Fibers, I think there hasn't been as much exploration into because they're tricky because just of the way that they're shaped, it's not like a defined diameter. So like if you have them long ways, obviously they're much bigger than if they were to like enter through a filter or some other substance like at their point, if that makes sense. So there's less investigation to that, but the fact that we're finding so many fibers definitely makes me want to explore specifically fibers in fishes more. So like, where might we find them? Are they even able to translocate? How small do they have to be, or what shape do they have to be in order to translocate into the blood and therefore other tissues? We are finding fibers in other tissues. They are definitely mostly concentrated in the gut tissue, so making their way into the gut and perhaps not always making their way into the blood and other tissues, but we are still finding some, particularly in the liver. So I'm not sure yet is the short answer to your question, but something that I'm working on.
Great, thank you. So Long, I have a quick question for you in terms of your spraying.
Yes.
How reproducible is it?
So based on our testing, I would say after optimizing the whole procedure, it is pretty consistent in terms of the material properties and quality, I think. So it is reproducible and scalable as well.
Okay, thank you.
Thank you.
Well, if there's no other questions, I know it's finals week for many of you, other than say Cal Poly San Luis Obispo, which still has what, another 2 or 3 weeks to go, I believe.
Yes.
And
please.
so, yeah, another 2 weeks to go. So I certainly we thank all of you for coming today, and I'll let you get back to grading or reading reports or whatever else that you're doing right now, and looking forward to a long summer. So thank you for attending, and hopefully you'll attend other events. We have one in this Friday on the EHR core research program from NSF. We'll have NSF program officer at 10 o'clock, as well as 4 speakers who are funded on the program. You can register for that. And we'll have another virtual cafe next month also. So with that being said, we certainly look forward to having you at another time in our future events, some during the summer. We'll take a little break in August, but we'll come back in September. And thank you, Monica, for posting some of our information in the chat box. Please, if you do have some time, please give some feedback, because it just only allows us to get better and to provide other types of activities and events to support faculty and students across the 23 campuses. So with that being said, thank you to our 3 speakers today, and to all of you, have a great day. Bye.